the file and said, okay, this is what I want to show in this paper, and he made a figure out of these pieces. They're not linked to any known record in a research record, any known date. I mean, it's just a hodgepodge. And so I think to this day he doesn't realize how egregious this was, but we certainly made findings. I still am stunned. Anyway, the consequences of this case was that after our oversight investigation, Dr. Venters confessed and agreed to the findings that we made. He had been resistant before that. And he was fired and lost his PhD degree. And the bottom line is that ORI can overrule institutional conclusions. The second case involved a graduate student in Francis Collins' lab. I think every one of you know who Francis Collins is. He's the head of NIH. And at the time of this happening, he had recently moved from the University of Michigan to NIH to be head of the Human Genome Project. He took over for James Watson. And he was busy setting up his lab, but mostly setting up his, in running the Genome Center. And this student moved to NIH with Dr. Collins, and in 1996, allegations were made by a reviewer of one of his manuscripts. And the misconduct involved 5 published papers, 2 review articles, 1 unpublished article, and a PhD thesis. And a GenBank submission was false. And so the penalties that were done by, I think, University of Michigan and NIH both carried out investigations. A 4-year debarment, which means, and I haven't really described what a debarment is, we call them exclusion. If it's a voluntary agreement with us, it's called an exclusion agreement, but it's equivalent to a government-wide debarment. And a government-wide debarment not only means that you can't apply for or be supported by NIH grants, it means you can't receive any federal funds, essentially. So if you're a physician, in particular, and you're in a misconduct finding involves a exclusion agreement or debarment, you cannot receive Medicare or Medicaid funds. So it's basically means that a physician becomes unemployable, which has happened rarely, but it has happened. And there were some papers retracted, and he ended up having both his MD and PhD degree denied, and he basically dropped out of science. So the consequences is that he was denied his degree and left science. I will say that this was a situation which was unfortunate because Dr. Collins, who's a remarkably capable administrator and scientist, was so busy running the Genome Center that he sort of didn't, for some period of time, wasn't really paying an adequate amount of attention to the student. And I think his quotes here was that he said, this case changed me forever, and it caused me to become more skeptical. This is, he's running NIH now. He still has his own lab, but I can guarantee you he's still watching. He's now watching the data of his students in his lab. The bottom line: misconduct can occur in any laboratory, and the PI is not automatically responsible for it, or aware of it, or how widespread it is. A little confusing. I apologize. But the PI is not automatically responsible. And as I said earlier, because in this case, in particular, there's no way to prove that he knew that the falsification was happening. If he had known about it and allowed it to happen, that's a different story. Now, the William Simmons case was interesting. This is one of my cases, I think. He started off as a technician and then became a graduate student at the University of Texas in a Health Science Center in Dallas. And I actually flew to Dallas to help them get established for on what to do about the investigation. And I remember distinctly sitting in the vice president's office, a big office, top of the tower they have there, and seeing this pile of boxes over in the corner, and saying that they said they'd be looking at all the data. And at the end of the case, I think what I concluded was that that's how they looked at the data. They saw the boxes and they said, we looked at the data, because they didn't really do very much. So, and they completed the investigation in a very short time. But Mr. Simmons at the time consistently got outstanding data using a complex cell assay. And I don't want to get into the details because it would take too long to explain, but it involved the use of radioactive chromium that was put into certain cells that had been treated in a certain way. The amount of chromium that was released when they mixed two cell types together, it's called a cell killing assay. There's a certain amount of chromium that's released from the controls that's called leakage, because the cells may not be very healthy. So you measure that as a background level, and then you measure the amount of chromium that's released from the cells, and that's the higher the level, either the more cell killing there was, or the healthier the cell, or the cells are more healthy. So it's a highly variable and difficult technique. And they were using transgenic mice and rats that generate the two cell types that were mixed during experimental conditions. And he was so good at this, fulfilling all the hypotheses of the geneticists and whatever, that everybody came to him for doing these techniques. And he finally got his PhD. He was there about 5 years, and went to Virginia to join a biotechnology company. And a new postdoc joined the lab from Japan, as it turns out, and he spent a whole year completely unable to reproduce Dr. Simmons' results on some of these transgenic mice. Well, as it turns out, Dr. Simmons lost his job in Virginia and was asked to come back to, in fact, he came back to Dallas, asked for a job, and they asked him to help the postdoc number 2 to set up the assays to make them work like they had before. Now, on a particular Friday afternoon, postdoc number 2 happened to notice this. Tubes in a rack, which are all set up in the same protocol. Make sure I get the right key here. Notice that there's double rows here, a single row here. Each of these would be a set of tubes for a particular combination of cells, controls, and other words. So these are like 4 sets of data, and they had several racks, and these are the data sheets that were next to these racks. All right. This is Friday afternoon. Actually, it's Friday early evening when Dr. Simmons had left for home. And postdoc number 2 said, "This is strange." And he noticed there was fluid in these tubes. These were tubes that normally would be used in a scintillation counter, or the counter for detecting chromium, but it wouldn't have been until after the cells had been mixed on the following Monday, and then they would measure the release of chromium. But stunningly or surprisingly, postdoc number 2 took a small aliquot from several sets of these tubes, and found chromium in them, and the number of radioactive counts was consistent with what the results would have been if the experiment had been done already and was completed. But this is Friday, not Monday. So what happens next is called a sting. You know what a sting is? It's where you set up somebody by misinforming them what they're looking at, in this case, and they get the results that is expected even so. So the lab chief was briefed on the situation by postdoc number 2, and he arranged with the animal facility to deliberately mislabel some mice and rats for Dr. Simmons to do some experiments the following week or two. And lo and behold, the results were exactly as though the cages had been labeled, and the results were perfect, but they were based on the wrong animals. So we knew that all that data was false. The problem is that right now, what do we have? We have 2 experiments that are false. None of them have been reported in the literature. None of them have been used for the basis of grant application. And there's concerns about what happened in the previous 5 years that he'd been in the lab, where all the data was wonderful. And the university stopped their investigation at that point and said, we're done, we got misconduct, he's fired. And so we decided to do some more work ourselves. And I asked for all the records that they could provide for similar experiments by postdoc number 1, Dr. Simmons, and by postdoc number 2. And I did a series of analyses of the data in terms of the kinds of things you'd expect to see with an assay that was inherently dirty. In other words, it has a lot of variability. And this is just one measure of the data from the 2 different investigators. These are the data from postdoc number 2, where it's a measure of the radioactive chromium that leaked out of the cells. It's just a background variable. And this would be a number that would come from one set of tubes that would be from one animal. And each of these is a different animal. And you can see how variable the values are in terms of amount of radioactive counts. And this is data by Dr. Simmons. Because why is this? Because he had his data figured out what he wanted, then you put in the same amount of counts into each tube. And that's why there's very little variation. He basically spiked the samples and got very, very little variation. And I have other ways of looking at his data in the same way. So it was pretty, and there were there's more evidence of misconduct that we found later. So the former graduate student and now postdoc was fired. He was debarred from receiving, and he agreed to this, from receiving grants for 5 years. 4 papers were retracted, and the bottom line is some data are too good to be true, and the truth will hopefully always come out. All right, the Mona Therashelm case was another complicated one in which Dr. Therashelm, assistant professor in the Department of Environmental and Occupational Health Science Institute at the University of Medicine and Dentistry of New Jersey. The complainant had been a collaborator with Dr. Therashelm when they worked together at the University of Rochester and moved to New Jersey, and they set up separate laboratories. And he also had a core facility for the university where they did confocal microscopy, which is what this issue is all about, stereology core lab, sorry. And he became concerned when they, actually as a co-author, had published several papers in which he had reported stereology data, and it finally dawned on him that she hadn't used his core facility to generate the stereology data. So he finally confronted her, and he made allegations to the university, and they thought he was not very credible, and they dismissed him at least twice. He finally came to ORI and made complaints, and we asked the university to do a more thorough job. And that process at the university uncovered some serious problems in which she was a very effective and aggressive falsifier of her record. She generated false records for New Jersey. She created a false witness and a fabricated